share my screen. Okay. Can you see my screen?
Yes, it's great.
Okay. Thank you so much, Carly, and for the warm introduction. It's already midnight here, but I'm not tired at all. It's so exciting to be here virtually, especially as receiving such kind words from and recognition from my former postdoc advisor. And I really enjoy my time at the Selecta Lab, and there's so much there. Uh, okay, I didn't directly work on the tension probe project while I was Emory, but I was really fascinated about the elegant and the huge potential of this technique. So when I became independent, I decided to work on explore this field in my own way. So today I will share some of work our group has been doing over the past few years. Uh, okay, before getting into the main part of my talk, I will let me briefly introduce my city and university. I started my independent research group at Wuhan University. Okay, it's a center of the start at the June of 2017, and I think after the pandemic, I think most of you probably know the Wuhan, heard about the Wuhan. It's located in the central China, nearby the Yangtze River, and I, uh, actually our university is actually considered one of the beautiful campus in China, especially in spring, when the cherry blossoms are in full bloom. And okay, so today I'm going to share three stories from my lab, all centered on how are developing the new DNA tension probe to study integrin force dynamics from ensemble to single molecule studies in living cells. Okay, first let me tell you about our initial motivation. And I note that lots of previous evidence suggests that the integrin force of the cells actually probably cover a much wider range than we thought. So the problem is that the early probes were not good at measuring the high force, especially when track them the force dynamics over time. So I borrow a schematic from Victor's paper. As you can see, different tension probe has their strengths and limitations. For example, the DNA tension probe, particularly for the DNA tension probe, uh, the conventional DNA hairpin probe are good for the real-time force tracking. But most of the time, they work in pretty narrow range. On the other hand, the TGT probe can measure high, strong force, but they are irreversible. So that means once the probe breaks, the DNA breaks, the force transmission stops. So we ask a simple question: can we design a probe that is reversible but also handle a wide range of the force? This is our initial motivation. Luckily, my first PhD student, Hong Yun, had a really great idea. He combines two existing design, the TGT probe and the classic DNA hairpin probe, and came up with a new version called the reversible shearing DNA tension probe, or just the RSDDP. Actually, a colleague helped me name this probe. And the cool thing about this design is that we can tune the uh, unfolding force of this probe in the two simple way. One is by change the GC content in the hairpin, like a conventional DNA hairpin. Another way, we can move the force loading side, similar like a TGT probe. So based on this design, the probe could has a much wide force measurement range, but more importantly, it's reversible. So we can use it to monitor the integral force with a wide range over time in the living cells. of course, we could use, we can systematically predict the unfolding force based on this formula. So then we made three versions of the probe. One opens around 17 piconewton. One at 45 piconewton. Another one is more mechanical stable, opens at 56 piconewton. So this will cover low, medium, and high force range. And then, with help from my colleague, we use a single molecule magnetic tweezers to calibrate this those force to prove to confirm that the peak force. So you will see that the result, the calibration, pretty consistent with our prediction. So the one thing I wanted to mention that we found that the probe, the shearing mode probe, has clearly hysteresis between unfolding and refolding. So just like a TGT probe, so the force reported by this type of probe is actually the minimum force that needed to quickly open the probe, not the transient force. Okay, so then to make sure the stronger probe is really stable, we test this by the magnetic tweezer. We apply a small force, a low level of force between 30 piconewton to 40 piconewton for a hold for like 10 minutes. We didn't see it open. So this experiment will make me more confident in the latest cell experiment. That means when we see the signals generated from this probe, this means it's a real tension signal from the high magnitude of force, not just the weak force hold for a long time. Next, we could functionalize this denotation probe with a different, or we could take this probe with a different force threshold and label them with a different fluorescent dye. Then we attach them together on a gold nanoparticle surface and creating what we call a dual quench system. Actually, this dual quench system originally designed by Yang when he was at Emory. We worked together, right? That's a great time there. And with this setup system, we can detect like we can do multiple channel image. We can simultaneously we can distinguish and track the different level of the integral force over time. Very interesting. We found that some structure, adhesion structure, with when the structure had a strong integral force, it also has a long longevity of the adhesion. So that we ask why the structure, what exactly of this strong integral force doing inside the adhesions? What's their rule? So then we did a simple experiment. We took a fibroblast expression GFP vaccine and let them spread on two types of surface. One coat at the TGT probe, and another one is on the reversible shearing probe. And you will see that clearly on the TGT probe, the focalization, the force cannot go beyond the 56 piconewton, and the focalization become more and more dynamic and smaller. But on the reversible tension probe, the adhesion was large and more stable. And then we diluted, we add the 2% of the reversible tension probe into the TGT probe and coating them on the surface. Surprisingly, that's enough. You'll see that the focalization quickly stabled and look very similar to the those on the 100% of the reversible probe coating the surface or fibronactin coating the surface. So this result suggests that a small number of the mechanically strong integrants could maintain the focalization stabilization. Then I asked my students further to confirm this. Okay, so then we designed another probe we call photocleavable sharing DNA sharing probe, reversible probe. We we add a tiny UV sensitive linker into the DNA hairpin loop, and then we could switch the probe from reversible to irreversible, and by using the UV light. So then we mix this probe, a small amount of this probe, only same before, only 2% of this fluorescent photocleavable probe into a sea of non-fluorescent TGT probe. So on the surface, like only one probe of every 700 probes was traceable and switchable. So at the early stage, when the cell spreading, everything looks fine. Like you could see a pretty stable focal adhesion, and we also see the tension signal, like a small single molecule level tension signal on this here. Then we did the switch. We exposed the sample to the UV light for a few seconds, and then that will convert the reversible shearing probe into irreversible TGT-like structure. What happened? You see that the force signal disappeared immediately. That means the probe had switched. And more importantly, some of the large focal adhesion quickly disassembled, as shown here. And so this will tell us even at the single molecule level, the mechanically strong probe still could keep the foundation structure stable. It's like a load beer structure beans in the building, and you don't need many of this. But without them, the whole structure become unstable. And okay. Okay, well, this pretty much of my first part of my talk. At the end of this story, I would like to point out that in the early version of the uh, reversible shearing probe, when the integral force went above 60 piconewton, the BHQ strand have some chance to fall off. We didn't realize at the beginning until one of the reviewer mentioned us. Fortunately, this probe is still attached to a gold nanoparticle. That's a second quencher, right? So even the strand, BHQ strand detached, the fluorescence still stay quenched. It didn't really affect our experiment, but still we want to fix this problem. So recently we redesigned this probe. We modify the BHQ directly onto the same DNA strand as a fluorescent dye. So that will give us has two advantages. First, the whole structure become fully covalently anchored to the surface, be more stable. Secondly, the BHQ are more close to the dye, so you will see that the quencher efficiency is even more. The quencher is more efficient on the new version of the probe. Okay, let's move on to the second part of my talk. I think many of us know that the stiffness of the ECM is a really important cue for cells because it could influence the many cell behaves and functions like cell differentiation, polarization, and migration. And to study the how cells sense their environment, we often use traction force microscopy, TFM, measure the cell force when they spread on a soft hydrogel to mimic the tissue. But the bigger limitation of the TFM is that the spatial resolution is pretty low and it cannot capture the molecular force, right? So we ask if we could use a DNA tension probe or other molecular tension probe to look at the molecular force on the hydrogel. Uh, So the problem, the idea is pretty straightforward, I think, right? The problem, the tricky part is unlike the cover slip, you could easily do the chemical functionalization on the glass, and the hydrogel is highly porous and heterogeneous. If you directly cross the DNA probe with the hydrogel, this probe will go into inside the hydrogel and generate a huge fluorescent background noise that will cover the real tension signal generate at the interface of the hydrogel surface. So how we solve this problem? Fortunately, another PhD student, Wen Xu, spent a few years to solving this problem. He pre-assembled the gold nanoparticle and the DNA tension probe into a large spherical complex in the solution first. This way, the probe cannot penetrate into the hydrogel. Instead, they prefer stay on the surface. Then he developed a chemical modification, shown here, and that will allow us to covalently attach this DNA gold complex to the hydrogel interface without affecting the hydrogel's original elasticity. On the top of that, we also put some fluorescent nanospherical particles with the hydrogel's interface. So then we could combine the traditional TFN with the DNA tension probe and get more comprehensive data like a molecule force map and the total traction force and the force orientation. So you'll see that to test this, we couch the fibroblast cells on the hydrogel interface and use a confocal to do a 3D images. And you will see that the tension signal reported by DNA force probe only localized at the interface of the hydrogel. Inside, you won't see any background from the background fluorescent in the inside of hydrogel. And okay, here's a video showing how we use a DNA tension probe and the TFN together to image the cell force spread on a soft hydrogel. You will see that the TFN could provide the total traction and the force direction, but it has a much but the DNA probe give us a much clearer and more detailed force on the force map. And we also use this method to image the protozoans in the microphage. Protozoans are pretty tiny and it's a very small structure with the actin at the center and surrounded by the integrins. You wouldn't see the protozoan tension signal using on the TFM channel because the integrin force are perpendicular to the surface. And but you will see this protozoan, the integrin force surround the protozoan with the DNA tension probe. And then after demonstrating this spatial resolution advantages of this method, we raise the next question. How do the individual integrins change if we tune the stiffness of the substrate? To start this, we use the TFM for test first. So the results are pretty consistent with the literature. When the hydrogel stiffness decreased, the cell spreading area for condition size become smaller, and the traction force become weaker, right? So the question is, at the molecular level, does the integrin force also change like this way? So next, we prepare a series of hydrogels with a different stiffness and functionalize their surface with two different type of DNA probe in a 1-to-1 molar ratio. One probe opens when the integrin force are higher than 17 piconewton. Another one is more mechanical stable, opens at 50 piconewton. We could obtain high quality molecule force map from all hydrogel sample. The weird thing is even on the soft hydrogels, where the cell can only form latent adhesion structure, we still see a lot of the integrins could open the 50 piconewton probes. If we perform a ratio analysis of this total fluorescent intensity from two different channels, you will see that to calculate the percentage of the strong integrins within the active integrins, so you will see that no matter how the stiffness of the hydrogel changed, the ratio remained very stable, close to the 40%. In other words, the ECM stiffness had almost no effect on the integral force, the magnitude of the force. This result seems counterintuitive, right? So we asked my student repeated this experiment in the room temperature. We also try other cells. Its results similar. So we also did to further validate this. We put the TGT probe on the hydrogels, the stronger TGT probe, and we did this experiment in the room temperature because at the room temperature, TGT probe become more mechanically stable. So you will see that the cell, the fibroblasts, were able to still be able to to open the TGT probe, the 56 piconewton TGT probe on both soft and stiff hydrogels. This further confirmed our previous observation. So the next question is, if the magnitude of the integral force doesn't change, what does the stiffness regulate, right? So we got inspired by a smart idea from Ron's PNS paper, and she used a locking strand to freeze the short-lived TCR force in T cells, so she could quantitatively compare how often the T cells scan the ligand on the surface. So we thought that could expand this idea, borrow this idea into our system for rigidity sensing studies here. So we also designed a small DNA strand into our system for rigidity sensing study. Then so if the integrin binds to the ligands at a different frequency on the different hydrogel sample, the fluorescent signal will grow up at a different rate. To test this, we use two types of the cells here, and one is a fibroblast, which like to move toward the stiff substrate. They usually call positive durotaxis. Another one is a glioblastoma cells, which strangely prefer soft substrate. They have been proved in the literature. They call this as a negative durotaxis. So we want to know, do their integrin scanning or sampling behaviors match their migration behaviors? Okay, so we add the locking strand and track the force signals grow over time on the different hydrogel samples on the different for the different cells. So here's a statistical data analysis here, and for the fibroblasts, in the most of case, and we found that the mechanical sampling effect decrease as a matrix stiffness increase, and suggests that the integrin binding frequency or integrin ECM interaction is unstable on the soft hydrogels. The binding frequency is pretty high compared to the stiff hydrogel sample. But for the glioblastoma cells, it was totally different. Their sampling rate increased with the stiffness, suggesting that the integrin binding frequency is fast on the stiffer hydrogel samples, and the integrin is binding more stable on the soft hydrogel samples. This result seems to match their migration behaviors. So of course, the molecule mechanism behind the dual taxis may far more complex than this. But one thing is more clear here. The integrin binding kinetics are more, I think integrin binding kinetics are more sensitive to the ECM stiffness than the magnitude of the integrin force. So we use the same approach to see how the T cells responds to the different stiffness. So we first try T cells using the TFM. So on the soft hydrogels, like 1 kilopascal, we could see clearly force signals from T cells. But on the stiffer hydrogel samples, 30 kilopascal, the signals disappeared. At the first glazing, it looks like the T cells apply less force on the stiffer hydrogel samples, but that's not real the case. Because the T cell naturally apply a very weak force as compared to the fibroblast. On the stiffer hydrogel interface, this weak force cause the displacement of the beads, fluorescent beads, are just so tiny for TFM to detect it. So then we switch to the DNA tension probe, the 12 piconewton DNA tension probe. So we'll see that on the soft hydrogels, the signals were pretty weak and discontinued. And on the rich sample, the signals will become stronger and concentrated around the CNAP. I think this suggests that on the soft substrate, the T cells may not the TCR force cannot reach to the 12 piconewton threshold, and this threshold has been improved by Yang and Chen Zhu's lab. It's needed for TCR activation. So then we wonder if the TCR scanning frequency change with the substrate stiffness. So we add the locking strength into the samples to see how the TCR force accumulate. So we okay. And so we saw that the TCR force signals accumulate much faster than on the stiff hydrogel sample, suggesting that the stiffness interface not only increase the magnitude of the TCR force, but also increase the TCR scanning rate. I think that's eventually boosting the antigen recognition and the activation efficiency. Uh, so I would like to use two cartoon to highlight these findings in the T cell. The T cells behave much more active and forceful when they interact with a rigid interface, but become sluggish and less responsive when they interact with a soft interface. I think it's maybe offer another perspective. The stiffness or rigidity could be a mechanical checkpoint in the T cell activation process. Okay, and let's move on to the third part of my talk. So far, most of my work has focused on study the magnitude of the cellular force. However, beyond the magnitude of the force, molecule force, the timing information of the force transmission is also very important, especially the force loading rate and the force duration. Because the force loading rate directly affect the rupture force of the protein the bond and the lifetime of the bond. For example, when the people measure the biotin-sirtuin bond, one of the strong non-covalent bond in nature, and using FM, you will find that the rupture force of the change significantly depending on the loading rate, range from 5 piconewton to 170 piconewton. And another key factor is the force duration, or how long the force could transmit it. This will affect the timing, determine the timing of the mechanical and biochemical coupling signals. And this part has been well demonstrated by Chen Zhu's lab. And also, force duration will also influence the dynamics of the protein-protein interactions and the protein conformational changes. For example, the titin I-27 domain has been improved, demonstrated it's mechanically stable using when people use FM to study this. And however, if you apply a very low level of force, just a few piconewtons on this, and hold for a long time using single molecule magnetic tweezer, you will see that you will see the unfolding process happened on here, as shown here. So this experiment show that the loading rate and the force duration pretty critical for understanding the molecular mechanism of the mechanotransduction. However, due to lack of the tools to directly measure these parameters in living cells, so our understanding of these parameters still limited. Like this review articles published like 15 years ago, point out that there's a wide estimation between different group on the loading rate, and the people have no idea about the force duration on the individual receptors. So, so that's why even this paper published like 15 years ago, this issue still haven't been resolved. Okay, so let's quickly go over the previous molecule tension probe. In the past, those digital DNA tension probe could only give us like a single data point on the force curve, right? So it's really hard to capture the full time dependent force transmission process. In order to measure the loading rate, we need a probe that at least could catch at least the two different force levels at the same time at the same probe, same data point. So the PG pack based fret tension sensor theoretically could do this, but in reality, the single molecule force signals usually fluctuate too much, and that will cover the real signals change caused by distance shift. So, that's why we want to develop a new probe to solve this. Two of my PhD students, Yu and Hongyun, said, why do we connect the two DNA probe in tandem? Actually, this idea is very straightforward, right? But the tricky part is how to synthesize this probe, because it requires multiple precise modification at the multiple size. Eventually, they propose a very simple and efficient synthesize strategy to achieve this probe. They link two DNA hairpin probes together, and the part of the probes are covalently bound. So that will keep the probe structure, the whole structure stable and reversible. Each DNA hairpin is labeled with different fluorescent dyes, and green for the low level of force, and a red one for high level of force. And so we also add a quench molecule at the junction of the two hairpins. So both dyes then quenched when probe is folded. And then we also add like functionalize this probe on 3 nanometer gold nanoparticles that will also help us prevent the photo bleaching, also as a second quencher. And this, when the probe under the force, the two hairpin will unfold one by one. If we could track this unfolding process at the single molecule level, the two hairpin, we could measure the time difference between the two hairpin opening and also give we could know how long the first hairpin lasts. This data will give us calculate the force loading rate and the duration. Since this probe is mainly used for measure the time-related force transmission, so we name this probe as a force chronoprobe. And we initially created two versions of the force chronoprobe that cover different force range. One, we use a double unzipping structure, which is sensitive to the 17 to 19 piconewton. And another one combines unzipping and shearing, and is sensitive to the 17 piconewton and the 41 piconewton. Of course, you could generate more other type, other probe cover more wide range. And before using this probe for the cell experiment, same as before, we use a single molecule magnetic twister to carefully calibrate them to confirm their peak force, unfolding force. We also check how the temperature and how the loading rate affect this probe. We found that the temperature and the loading rate do not change to the unzipping structure too much, but they do some effect on the shearing probe. Those testing results will help us to understand the possible error range when we use a different probe in the different experiment. So in the cell experiment, we dilute the probe to the single molecule levels and mix them with a non-fluorescent RGD peptides to coat on the gold particles, and that will create a spatial sample for the single molecule force image. Then we use a turf microscope with a tool to capture the images with a 2 EMCD camera and with a very high frame rate. And okay, this videos shows a typical single molecule force image. Unlike ensemble experiment, at the single molecule level, we can actually see the blinking of the force. And so by analyze each point of this spot, we could create a time-resolved fluorescent trajectories, which lets us precisely measure the loading rate and the duration of the integral force. Actually, in fact, this probe could report on the force magnitude, position, duration, and the loading rate all at the same time. Right. Also, each video contains thousands of data points. So then we could perform the statistic data analysis on the loading rate and the duration. And use this method, we could we found that the loading rate generally stays in a pretty narrow range, about 1 piconewton per second. And however, the force duration varies a lot. It depends on depends on cell type and the cell spreading stage, the adhesion size. For example, at the initial cell adhesion, the force duration is about 10 to 40 seconds. When the focal adhesion become more and mature, the force duration increase. At the same time, the loading rate decreased. And about actually it's also I would like to point out that almost at around the same time, TJ Han lab and the Carly's lab also report a similar loading rate values that will confirm these values are really reliable in living cells. So when we look at the single molecule fluorescence signals, we notice that even within the same adhesion sites, not all the signals green signals show up in the red channel. That means not all the integrants apply enough force to open the second probes. So some only only generate force, weak force. This is not a surprise. What a surprise that the weak integral force are more dynamic compared to the strong integral force. So we divide the integrants into two groups based on the force magnitude. And you will see that this class 1 integrants, they only apply weak level force. And the second, this type of force integrants could open both of the hairpin probe. And we found that the integrants apply strong force and last longer. This is a little weird, but maybe multiple reasons to explain this. Probably much we expect for catch bonds. Because the force on the integral ligand bond increase, the bond stability also increase. Yeah, maybe you may have other idea to explain this. And then, as you can see, this approach allows us to monitor the dynamics of the force transmission in living cells. This single molecule force fluctuation curves remind me how the doctor check our heart beating curve by look at the heart based on the beating curve, heart beating curve. Since the force transmission of the integrants in cells is also regulated by some key cytoskeleton or foldation proteins. So we wonder if we could determine these specific proteins functions or the role of these functions on the force on the mechanotransduction process based on by observing their changes in the force fluctuation curves. So then we previous study has shown that vinculin plays a key role to connect the integrin tannin complex to the cytoskeleton and help the cells engage and disengage to their surroundings. And however, how the vinculin impacts the integral force dynamics in the living cells, particularly at the single molecule level, is still not fully understood. So we create a vinculin knockout cells, and you will see that without vinculin, the focalization formation is not affected. But after knockout of vinculin, the integral force drop a lot. Let me suggest that the vinculin could sustain the force magnitude. And now let's look at what happened at the single molecule level. In these videos, you can see that clearly see that difference in force dynamics between wild type and vinculin knockout cells. If we do quantitatively, you'll see that the force become more dynamics in the vinculin knockout cells. And if we do the quantitative analysis of each point, the result will be more clear. Without vinculin, the force transmission become more unstable and with rapid and short-lived force signals. In the vinculin knockout cells, like the force duration will drop from 100 seconds to the 16 seconds. And at the same time, the loading rate increase. So this suggests that without vinculin, the integrin cytoskeleton linkage become unstable and leading to quick force application and release. Okay, in fact, the vinculin functions through conformational changes that expose the binding sites, especially the head domain contains actin binding domain, binding sites, and the tail domain has actin binding sites. So to explore this further, we reintroduce two mutants into the knockout cells. We found that the actin binding deficient mutant could partially restore the integral mechanical dynamics to the wild type cells. However, the tiny binding deficient mutant behaves like similar like knockout cells. This suggests that the tiny domain, only the tiny binding domain of the vinculin contribute more on the maintaining the integrin force transmission process. Altogether, this findings this probe acts like a cellular single molecule force spectroscopy tool that allow us to study the functions of the specific protein domains and the mutants under the mechanical force in living cells. So of course, there's many other cytoskeleton and membrane proteins that could impact the force dynamics. So some proteins may only influence the loading rate, while others could influence the force duration. I think use a similar way we could test and quantify their changes to analyze how they contribute how they tune both force duration and the loading rate. I will show more and okay and I think pretty much I my talk. So there's a summary of my talk. So in the first story, I introduced the DNA-based reversible sharing tension probes, which we use to identify and track a different magnitude of the integrin force in living cells. And in the second story, we tell how we functionize a DNA tension probe or other probe on the soft hydrogel interface to study the cellular rigidity sensing. And the last part, we developed a new probe that allow us to quantify the temporal dynamics of the force transmission in living cells. Also, this method works like a single molecule force spectroscopy tool in living cells that will help us to study the protein functions uh, through detailed mechanical measurements. And okay, that's I want to thank my students for their hard working at this, and then my collaborators for their help with the single molecule magnetic tweezers and the cell knockout experiment. And this picture, this one is my current group, and this one is when I start my lab. At, actually, these three stories I shared today were carried by those three students who now already graduate. Hong Yun is currently is a postdoc at Penn State University. Uh, and Wen Xu now is assistant professor at the medical school in China, and Yu Ru is currently applying for a postdoc position in U.S. In Harvard. Unfortunately, due to the current situation, her application is still on hold. So she's still working in my lab at the moment, and. Okay, that's all. Thank you all for listening. I will be happy to answer any questions you may have.
Wonderful. Thank you so much. That was an incredible amount of work and a lot of progress. We have about 10 more minutes, so I'd love to open it up for questions. You can either put a question in through the chat or just unmute if you'd like to ask your question.
Hey, Tim.
Oh, hi. This is Tim Springer here. So
Hi.
hi. Good to hear you talk. I have a question about the length of time that bonds maintain the force and the strong and the weak bonds. And I think could a lot of this and then you finished with very interesting experiments showing that vinculin has a major force on the strength of the bonds that are formed. So I think that is it possible that when you're measuring the differences on stiff or soft substrates, that some of the differences you see are not due to the integrin ligand binding rates, but are due to cytoskeletal effects on maintaining the force, maintaining the connection between the integrin and the ligand.
Yeah. So that's a great questions, and so it's also in the glass, and you see that the strong integrins has a longer lifetime. I think the education size become more and with more mechanical strong integrins are likely to trigger long-lasting biochemical signals. So they will recruit more tannin, weakening actins that will further helps to stabilize the focal adhesions. But from the hydrogels, because we cannot functionize the last probe we developed on the hydrogel samples to do the single molecule force image. So we didn't know the time on the different hydrogel samples. We just need to guess how what's the time information on the different samples. That's also puzzled to me as well. So one explanation is that on the soft hydrogels, probably the force, integral force, when they reach the certain level, the rotation size may be more, maybe move along with the integrins due to the soft of the hydrogels. And that could prevent the tanning from unfolding. Then that's essentially for the downscreening signaling. That's why the adhesion size cannot grow and stabilize. I don't know if I answer your questions or your concern.
Yes.
So maybe if I interject for a second, I think the question is you're measuring force duration, but you don't know about binding, the integrin binding. And so when you see the loss of force, it could be due to integrin ligand dissociation, or it could be dissociation of the cytoskeleton from the cytoplasmic domain of the integrin. I think that was.
Yeah, Kayla, that's a good way of rephrasing it. Yeah, I think that the cytoskeleton has a major effect. So I think when we think about the role of substrates in modulating this, I mean, that has the role of the cytoskeleton has to be included in addition, say, to the rate at which the integrins are scanning the ligand. I mean, so our own experiments on single molecules, show that once the ligand binds to an integrin, it converts in 70 milliseconds to the extended open conformation that then could bind talin and then interact with the cytoskeleton. And it can go the ligand can dissociate from the low affinity state in 500 milliseconds, but there's also a very rapid interconversion between the different conformational states of the integrins, in much less than a second. So I think that a lot of the conclusions about force exertion have to not only take into account, what the integrin ligand binding and dissociation is contributing, but also what cytoskeleton binding is contributing.
Okay, I see. Thanks for the comments. You mean the integrin ligand interaction connecting before the force application. Yes, in our case, we don't know how when they interact together. I think probably we can combine the two techniques together. Probably could answer your question.
Great. Yeah, go ahead, Alex.
Sorry. Zheng, that was a really fantastic talk. Looking at the data for your chronoprobe, one of the best things about it is its reversibility. So you get to watch forward, but also backwards and back and forth and back and forth. And looking at all the long traces that you showed, or most of them, it seemed like you would have flickering. But a lot of times it was just the red going down and the green would stay up. I don't know if that's true or just a false observation by only seeing a few of them. But the thing that's kind of intriguing about it is if you look at your magnetic tweezer, the two forces of refolding, so red going down and red and green going down, are actually very close together. They actually looked within 5 or 10 piconewtons, and you had a very similar loading rate for that. Do you know why that is? Is there any information that you can get from this red flickering up and down compared to red and green flickering up and down and those kinetics?
That's a great point. Actually, let me show you more clearly video. Okay, like this one. You see the red signal and the green signal always disappeared at the same time. So that means the linkage between the integrin, ECM, actin, tannin breaks at a certain point. That the integrin force is released, right? So in the most of the time, you will see that in most time the force just increase, continue increase until the linkage breaks. And for the magnetic tweezers,
Yeah, so this was the one where it stuck out, where I saw a lot of red going down, but not green. Both going down is just like a release, but one of them going down at a time is like a
I think
slow release.
both. I think the both going down. You see here.
Yeah. There, right there at 200 seconds.
Yes, that means the integrin, the linkage of the integrin and the cytoskeleton break.
Oh, is it both and it just happens so fast that you can't see it?
No, I think they happen at the same time.
Sorry, yes, at 150 seconds, they both happen at the same time, but at 200 seconds, it looked like red went down. But I just don't know if the temporal resolution's so fast.
Oh,
that
there's, I think the red curve just cover on the top of the green one. So you didn't see that.
Okay. And is that the case most of the time, where it's always they both go
Yes,
down?
yeah.
Okay.
Yes, exactly. Here, like this one. You see?
Okay. Yeah, like that. And it's clearer because there's a little bit more time. Is there any information to be gained with how quickly it grabs onto a new one?
I think based on the single molecule magnetic tweezers, they're refolding pretty quickly. Let me see. You see we apply 1 piconewton per second. So we see the probe refold at within a second time scale. Like this. Just one data point, you see the probe refolded.
Yes, I was talking about the interaction once it lets go. Sometimes it grabs on really quickly and sometimes it grabs on again slowly. And if there's any kinetic information that you could get there. But yes, I agree with you. The refolding does happen pretty instantaneously. Thank you.
Even the unfolding is also pretty quickly.
So maybe we have time for one more question. Sorry, I know Zheng's had his hand up for a little bit of time. Zheng, if you want to ask your question, thank you.
Yeah, hi, Zheng. It's a great talk. So you had in the second part using hydrogel to look at the stiffness of the hydrogel on the forces the cell generated, right? And I don't remember whether you also have the loading rate as a function of the matrix rigidity. But the probe itself, it's also a spring that is in series with the hydrogel, right? So I wonder, what are your thoughts on the property of the probe that affects the measurements?
That's a great question. One of the review also asked these questions.
I'm
It's
not.
pretty challenging. I know. It's pretty hard to do the calibration single molecule magnetic tweezers on the hydrogels, because when we put the beads on the hydrogels, it will generate lots of interference pattern. What we did, let me show you. I have the okay, let me show you. I have the data. Sorry. We actually, you see that the unfolding force only depend on the free energy of the GC content of the hairpin. The rigidity of the substrate will not affect the critical force. To prove that, we create we put a different handle, DNA handle on the probe, that to mimic the hydrogels. Even very long handles, you see that the unfolding force is still same.
I think what I was wondering isn't the probe or the hydrogel property, is the cell. The cell, when it sends the probe property and the hydrogel property, it would respond by maybe exerting more or less forces or higher or lower loading rate. And so what your measurement was what the cell did, right? The probe is just reporting what the cell did.
Yes. I think even the
Highly so.
Yeah, you see that this probe only open at the certain force threshold. I don't think the substrate will affect the unfolding force. I know that there's that's our point. It's this experiment.
No, I'm not saying that the probe would open the different forces because of the hydrogel. I'm saying the cell sense the different property of the probe and the gel, and as a result,
Okay,
different
I see. I see your point. Could be, but on the soft hydrogels, you see that the cell only form the latent adhesions. That's the only way we could determine the cell is like on the soft or stiff hydrogels based on the fluctuation size and the spreading area. Yeah.
Okay, thank you.
We made yeah.
Okay, great. Maybe I see maybe Vakisha has one question. Is this Vakisha the same question you have in the chat?
Yes, it's the same question.
All right, maybe last question, then we'll end here. Okay. We don't want to keep Zheng any later. It's already 1 a.m. for him.
So very quickly, when you were doing the microscopy part, how were you able to differentiate between the breathing versus a very transient pulling from the cell?
I mean, on the single molecule image.
Yes.
In our experiment, most of the time that is the force is detectable, like a few seconds. So we didn't see we did see some transient force, not too much here. When we perform the statistic data analysis, we include this point. Yes. but I don't think they contribute more on the force duration and the loading rate values.
Okay. Awesome. Thank you.
Because the frame rate we connected is like the time resolution is in this case is 50 microsecond.
Gotcha.
So pretty fast.
Okay.
All right, Zheng. Thank you so much. I appreciate the time you took, and you know, congratulations on all these really nice results. And thank you, everybody. We'll see you next month.
Thank you. Thank you all.